In this video, we're going to take a look again at this process of how DNA is used inside of cells and what can go wrong, but we'll focus a little bit more on the process of how we can actually get DNA repaired, and then finish up taking a look at some methods that people have used to actually identify mutagens that are out there and how they can be used inside the lab. And so if we look at this process, if there is damage that occurs to your DNA, whether it's during DNA replication or just during the normal cell cycle when it's sitting there using the DNA, the cell wants to be able to both detect those mutations that might have occurred in the DNA, but also they want to have the ability to repair the DNA so that you don't have to worry about those mutations possibly ending up in a constitutive gene, which means that now you've got something like, say, an enzyme for glycolysis that no longer functions. That cell is going to lose out on glycolysis, and unless they can have an alternative pathway for getting energy out of glucose, they're more than likely going to die. And so if we take a look at the two major methods that cells can use with their DNA repair, one of them is photoreactivation, which we looked at a little bit before in exercise 20 when you exposed your cells to UV light. And that UV light is non-ionizing radiation, so it's going to come in there, and instead of causing breaks in the DNA like you would get with something like the radiation coming off of a nuclear reactor or uranium or something like that, here the UV light is just going to excite an electron in these pyrimidine bases, and usually it's going to be between two thymine bases, and that electron is going to start jumping back and forth between the two bases. And so this is going to cause that DNA to basically kink or bend as you get the formation of the dimer. Again, when you think about this process, electrons jumping back and forth, well, here you're looking at something that would be analogous to something like a covalent bond there, where the electrons are jumping back and forth between your atoms, but now they're jumping back and forth between these two molecules here. And so this process can result in a change in the shape of your DNA. And when that DNA is read and being used by the DNA polymerase, because there's this bend right there, it'll often result in the DNA polymerase just come shooting along, and it'll skip those two bases right there. So now you've got a double point mutation, and that's going to cause a frame shift that'll throw everything off again. And what happens is there's an enzyme floating around in the cells that's known as pyrimidine dimerase or photolyase, depending on which paper or textbook you're reading. And this enzyme is going to be floating around, and it'll bind to the DNA and scan along it. And when it runs into this dimer, the enzyme is going to stop there. And if it is daytime, then the visible light that is hitting the cell is actually going to get absorbed by a cofactor called folic acid that is built into that enzyme there. And that energy that is absorbed from the visible light is going to be used by this enzyme right here to basically move those electrons apart, and that'll restore the shape of your DNA, and you've got your normal DNA back. So really, this is a low maintenance kind of thing. It's basically scanning it, and when it finds that error, it forces the bases to separate so that your DNA is back to its normal shape. Pretty simple and pretty easy. But what do you do during the nighttime? Because remember, this requires visible light. And if a cell gets exposed to a whole bunch of UV light in the afternoon, they still need to be able to fix this change to their DNA. And so they have a separate mechanism out there that is called nucleotide excision repair. And here you've got another enzyme complex, like our photolyase out there, that binds to the DNA, and you can almost look at it like a donut, and it's scanning along the DNA, and it runs into this bend or the kink in DNA that causes the complex to pause right there. Well, then this is going to result in an enzyme called a nuclease to come in here, and that nuclease is actually going to make physical cuts in the phosphodiester bond here, where you have your sugars and your phosphates there. And then a helicase enzyme is going to come in and basically break those hydrogen bonds and separate this chunk of DNA that is now going to come out of your DNA, and that's going to leave a gap in your DNA. You've still got your 3' hydroxyl end over here, you've got your 5' phosphate over here, and if you think about this, this. This is exactly what we run into during normal DNA replication on that one part of the leading strand, or over and over when you look at the lagging strand with Okazaki fragments. You're going to end up with your DNA where the RNA primer is removed there. And so the same enzymes are going to come into play here. Our DNA polymerase 1 is going to take that existing 3' OH and use it to add your bases that line up by hydrogen bonding between the complementary bases. You will get hydrolysis of those nucleotide triphosphates to provide the energy for your phosphodiester bond to be synthesized there. At the very end, you run into that same problem that you don't have the DNA polymerase ability to connect that last part with the 3' OH and the 5' phosphate on these two pieces, and so the same enzyme is going to solve that problem. DNA ligase is going to come in here and it's going to seal that sugar phosphate backbone right there, and now you've got your two strands of DNA back to their normal thing, and you've removed the damage that occurred there. So now we're going to switch gears a little bit and look at the second part of this, and this is how people have found that they can actually use mutations in the lab to start looking at some of the genetic characteristics of these cells here. And we'll take a look at how you can isolate mutations in biosynthesis genes that are out there, but also how we can use these mutant bacteria to actually identify mutagens that are out there. So chemicals that might end up in your food, you would probably want to know if those chemicals are going to have a mutagenic effect on DNA that is out there. And so we'll take a look at an example, and here we have one of the sort of standard things that people were doing, because they could grow up cells on complex media, and that complex media would provide the amino acid histidine to those cells. But if you have a mutation in that histidine biosynthesis gene, if there is a mutation that disrupts the enzymes involved in making this amino acid, then those cells are going to die out. But you can supplement your media by adding this histidine there. And so this is a great system. We can go in there because we can start using this to identify things where there's a mutation occurring. Here we've got our polycistronic message that would be transcribed from our hist gene right here, and then that message is actually read by the ribosomes, and in this case it's polycistronic because there are actually 5 different coding regions built into the message. So there's 5 different AUG start codons here, and the ribosomes, as they're chugging along reading this message, can actually end up making 5 different enzymes that are going to take precursor chemicals out there and modify them into our final amino acid histidine. Well, imagine what if a mutation occurred that disrupted this AUG right there. And now you've got a change where that AUG has changed the codon to an ACG. So just a simple point mutation occurred right there at the equivalent location where your start codon is in your gene. Well, if you think about this, now you're no longer going to be making this first enzyme in the pathway. So even if you have the precursor right there, you're not going to be able to turn it into the first product that is used by the second enzyme, which means your cells are no longer going to survive unless you supplement them with histidine. Complex media, it's already in there, but remember, we've got the other type of media out there. We've got our defined media where we know exactly what kinds of chemicals are added, and we know the exact amounts of them in there. So if you look at this process, people realize I can take this and take my wild type cells out there and try and get some mutant versions of this coming out of those cells there. And if we take a look at this figure, you can see how you do this process. You basically take your population of cells and grow them up on something like complex media where they've got the histidine there, and you dilute them down to where you have single isolated cells that are going to grow up to form colonies. And then you use a really high-tech method here. You take a piece of velvet and you put it around this ring and press it onto your plate, and now you've got your colonies with those cells from each separate colony there on the velvet, and you stamp it onto some media here that contains histidine, and then you replica plate it or stamp it onto some media that is your defined media that is lacking histidine. And then you let these plates incubate and you compare the pattern of growth onto both of these plates here. And if you look closely at this, you can see there is one little spot right there where this colony is missing. And so these guys grew here in the presence of histidine, but they could not grow in the absence of histidine. And this means we can go in here and scoop these cells off and transfer them to a new plate with histidine added. And now we've got a histidine auxotroph. We have some cells that don't grow on media that are lacking histidine. And what people thought about is, can we actually use this to start identifying different kinds of chemicals or treatments that can actually result in an increase in mutation. And the reason why they're doing it is kind of summarized in this and what's known as a reversion mutation. So imagine you started out with your population of what are known as wild type cells. They've got the normal AUG. A mutation occurred in that first codon right there that knocked out our enzyme, so those cells could no longer make histidine, so they are what are known as His- cells. Well, what if you had a mutation that occurred in that second position of this codon in the DNA, and it actually caused it to go back to the normal? So you can think of this almost like a silent mutation between the wild type and this revertant that is out there. There is no difference between those because now you've got your AUG for your start codon. And so if you think about this, if we start out with this little guy over here, they're all going to die, and the only way they're going to be able to survive is if they have a reversion mutation. So we can use this system to start testing different chemicals out there to see if they actually cause or increase the number of those reversions. And this is known as the Ames Test by a professor out at UC Berkeley who came up with this to try and test chemicals to see, are they actually mutagenic, and these things could potentially cause cancer inside of us if they change the DNA in some cells. And he used a mutant version of Salmonella. He made two separate types of mutation. One of them is that auxotroph mutation in our histidine biosynthesis gene, so that your cells cannot grow in the absence of histidine, but also he deleted these genes that we saw at the beginning of this that are responsible for DNA repair enzymes. Because if you have a mutagen that increases the number of mutations and then the cell fixes it, you're never going to be able to see that. And so we take this thing and we set up a control plate where you've got minimal media lacking histidine, and you can throw in a control. In this case, you could throw in something like water or whatever you're diluting your chemical in there. And then you take that and you incubate your cells with this, and you plate it out. But some people argued that inside of our bodies, our liver will actually have enzymes that take chemicals that are toxic, and they convert those chemicals to make them less toxic, which could potentially make them mutagenic. Or your liver enzymes take mutagenic chemicals and they modify them so they are no longer going to cause problems. So how do you get around this? These are just bacteria cells. And so his solution was to actually take and grind up some rat livers and then extract the enzymes out of there and throw it into the tube, because those enzymes might modify the chemicals that are out there. And so you incubate this stuff and you plate them out and you look for the number of reversions that occurred here. These are spontaneous ones that would occur just from DNA polymerase causing mistakes. And this is how they calculated that error rate out there. So now we've got this system set up and we can do the same kind of thing. Here we have our control plate over here, and this is giving us the number of background or spontaneous back mutations that are occurring out there. And over here you do your experimental plate, and the only difference with this one is you can throw in your chemical that you're testing to see if it's a mutagen. And if we look at this, we get an increase in the number of reversion mutations. So we would see an increase in the number of cells. And so if you think about this, if your test plate has an equal number of colonies to your control, you would say that chemical is not mutagenic. It did not cause an increase in the number of mutations because we didn't see an increase over our background level. If you have more colonies on here, then you're going to say that chemical is mutagenic. And if you have no colonies on here, you're looking at a chemical that we don't really care if it causes mutations because it's toxic. No cells could survive in the presence of that chemical there. So the Ames test is kind of a cool way of identifying these mutagens that are out there.